accepted, reviewed, and authorized, they will continue to receive higher priority than full authorization. So, and this should be for obvious reasons. But we're still looking to expand testing capability and converting any eligible full authorization is usually and not expanding testing and close our reviewers away from new applications. Providing still prioritizing new applications. However, some are already coming in and anticipate authorizing those. Next question. Are there tests being developed to follow through on the viral load and associate them with disease activity for COVID-19, such as long hauler patients? Of course, I don't believe that universally the long haulers still shed virus. And as far as viral load tests, which would be fully quantitative tests, if such tests is something that the developer would do, that would involve the use of calibrators, a link to the international WHO standard. However, while we're considering such submissions, it does remain unclear to us what the clinical usefulness of such viral load assays would be. Certainly see, lots of research potential for, somebody doing purely research and wanting to try to figure out if viral loads mean anything. We continue to monitor the literature. We also continue to monitor the CDC recommendations, other professional and lab associations, and to date, they uniformly discourage the use of viral loads. As far as long hauler patients go, there's likely a need to identify these patients up front, if possible, or to assist in their diagnosis. We do note that there are publications that a patient who has been suspected of long hauler disease and didn't have a diagnostic test performed, and they may have been exposed and developed an infection, because non-symptomatic, asymptomatic individuals can suffer from long hauler, or someone may have been symptomatic and simply didn't get and had a mild disease and didn't get a diagnostic test. So there are publications and clinicians can do their best to use serology tests to identify whether those patients have in fact been previously exposed. Certainly come in handy, and there are a number of developers who are looking at other biomarkers to try to assess risk and potentially diagnose a long hauler, in addition to other clinical hallmarks of that disease. And so we do remain open to those clinicians and professionals and developers for that. But we have not, other than for IL-6, which predicts potential for ventilation. We've authorized 3 IL-6 assays. Other than that, we have not authorized any other sort of immune response assays for use in patients who suffer from more severe consequences of the virus and or lockdown. And moving to the next question. Given that no flu samples have been available during the last flu season, and currently circulating, not much circulating flu, as I can tell, based on the CDC flu report this morning, is it possible you can provide swab samples to prepare for an EUA submission for a rapid lateral flow test for SARS-CoV-2 today and previous antigens, in order to obtain a high-security swab sample? The swabs will be supplied with BTM samples and blind blinded for testing at the testing site. So we do not recommend this for a number of reasons. One is we actually don't recommend VTM for lateral flow use for SARS or really any other virus. We've just seen for under an EUA, we've just seen so many challenges with VTM, and VTM has been, some sample types for some assays have needed to be pulled back because of false positives with VTM. And then the other thing is because there are cleared, fully authorized EUA, 3D, RC, and other viruses that are available to use. We are not recommending the use of 5 samples for fluid and fluid, and that would be especially for antigen pathway. So again, I mentioned at the opening that if you want to have a panel test and you're not seeing active disease for some of your targets, if you can find a way to do the validation for the targets where you do find such samples for antigen tests, that would be awesome as long as those results aren't reported out to the clinician of the patient. Okay, Chris, I can go over to you for the next.
Thank you, Tim. The next question is the FDA considering EUA submissions for saliva-based multiplexed fluid COVID molecular tests? And if so, is that one question permitted and which template should follow? So the data supporting flu testing using a saliva sample is limited. Also, there are no cleared devices or EUA tests which are authorized for detection of a saliva sample. Our recommendations are to establish an EUA claim. We recommend using a molecular EUA template which notes testing date of the samples. Unfortunately, some of these samples are not available in saliva. So in lieu of that, we recommend a cleared sample clinical study establishing performance of a saliva sample compared to nasopharyngeal swab samples. To support a flu claim for saliva, it should be done prospectively. Furthermore, validation using fresh, prospectively collected positive samples will likely be challenging as incidence of flu infections remain low. So this is really a data-driven question. And the suggestion here is that the data is still involved in saliva. The second part is home collection, and that is something that is likely possible. If you have a number of home collection kits, and that specific question can be answered with the box or if you have a back to you, Tim.
Thanks, Chris. The next question actually is also saliva. This question, my question is in regards to the validation of saliva-based SARS-CoV-2 tests for EUA. Given the rapidly expanding body of evidence that testing of saliva specimens for SARS-CoV-2 is just as sensitive and often more accurate than usual swab specimens, does the FDA have any plans to consider saliva comparator assays for validation of new point-of-care diagnostic tests? The requirement to collect 2 specimens for each patient in a care setting is a critical barrier for validation of saliva-based assays. Given the importance of approval for saliva-based diagnostics to help facilitate testing compliance among young people, is the FDA considering a shift to allow highly sensitive saliva-based comparator assays to validate point-of-care tests? So in our view, based on the data and the review of literature, we would not agree with the assessment of this independent question as far as saliva being a better sample type than one of the nasal swabs. Saliva remains a very challenging sample type. Obviously, it's challenging because we don't know what flu or other respiratory viruses are able to be detected as accurately or accurately enough be utilized in an assay. Um, so we do currently still recommend that you test these drugs if you want to utilize or validating study compared with third CT swab using a highly sensitive molecular test. Chris, over to you. On part 3, I wasn't sure about this with you. Chris, you just go with what you need to go ahead. Okay, thank you.
So this question is a section that kind of rest on the order. This is concerning the use of the BioFire respiratory panel 2.1, the part 2.1, the method for anterior nasal swab appointments. It was previously stated that this would be an acceptable comparator for anterior nasal swab. Acceptable for anterior nasal swab appointments. Candidate assay to nasopharyngeal swab appointments with BioFire 2.1. This alternative to supporting anterior nasal swab appointments. So it was also stated that using the BioFire 2.1 as a comparator method, there may be additional information requested to ensure your SARS-CoV test has been validated with samples and the environment. And this is again in reference to that particular test returning a CP value, which is not equivalent to a CT value, just to put it close to speed. So going to the first part of the question, can we elaborate on what additional information would be required to demonstrate that the clinical study includes low viral load samples? So we do recommend providing a sufficient number of low positives. This is all covered in the template. These low positives should be characterized by a highly sensitive RT-PCR EUA test, which offers a CT score. For EUA, we have accepted study data that uses the combination of comparative methods and read testing. Answer the second part of the question. The second part of the question is, would it be acceptable at the completion of the clinical study to test not only positive samples but the real SARS-CoV-2 test that is CT? Again, we have accepted providing multiple comparative methods to establish performance. So that's something that would be open. I would suggest again discussing with us first. A lot of details which we so generally yes, but let's not exactly. The second question is developing a multi-analyte test using BioFire of the 2.1 as a comparator. Is additional information required to demonstrate that samples with low viral load were tested for other targets such as influenza and RSV? So I would say that the pre-selection of archived positive samples should represent a range of viral load or CT values, including the positive samples. We expect that the viral load of flu and RSV samples will be reasonably distributed throughout the measuring range. Part 3, if establishing performance from anterior nodes by comparing to a clear BioFire 2.1 imaging survey. What are the performance expectations? So again, this is covered in the template, and I believe that you should definitely check the template, but it would be for PPA of greater than 90%. And an NPA of greater than 95% in comparison to FDA completed. Would it be acceptable to instead establish anterior nasal swab claims by comparing nasal swab performance on the candidate test to nasal swab results in BioFire? This is also goes on. It says provided that the equivalency study for nasal swabs is conducted. So it is generally acceptable to perform an anterior nasal swab to anterior nasal swab comparison for EUA. However, in this case, only the nasal swab claim will be granted. We recommend further validation for other respiratory samples. So believe the intended use for the biofire does not include anterior nasal swabs. So they would suggest that this person again write up a detailed proposal, explain exactly what they would do, and the justification. We can consider that going forward. Typically, we want to see an EUA test used on label. That's what. Okay. Thank you for anything there for that one. And it looks like I will take the next one as well. Has the center, CRH, issued any specific guidance or suggestions on seeking classification for COVID assays and EUA? And is the biofire the appropriate predicate for COVID? So we are currently working on a transition plan for devices offered through the EUA. This is included in the center's guided priority list. Since biofire was granted a de novo, they are currently the only legally marketed predicate for classification. This is, of course, until subsequent tests are cleared through the 510 pathway. FDA recommends that we tend to pursue the 510 pathway, which is submit a pre-submission to discuss proposed validation strategies for third method. If you are proposing intended use, which is different from biofire respiratory plan 2.1, the discussion is recommended. And moving on to the last question. This company is working on a prescription at-home molecular SARS-CoV-2 influenza test. And the question is, does the readout displayed to the home user need to distinguish between influenza A and influenza B? Or could the readout simply say whether the sample is positive or negative? This is a question that is quite thoroughly discussed. And we do recommend that influenza A and influenza B are differentiated as a test result. And this is based on external opinion and discussions with flu experts, and has consistently been recommended for previously cleared 510 tests. With that, I believe we are done with the submitted questions. Let's move on to the next phase.
Yes, let's open it up for live questions.
Thank you both for all of that, and thank you again for your email questions. So please make sure if you want to email questions, you do so before the next town hall, which will take place in 2 weeks. Now we'll switch to the live question, and as I stated earlier, when I call on your name, please promptly ask your question. And as a reminder, we cannot answer any questions as they relate to specific device submissions. So with that, the first question that I'm going to go to is from Anthony. So Anthony, please ask your question. First, you'll need to unmute. I'll move on to Autumn. So, Autumn, please ask your question.
Hi, can you hear me?
Yes.
Okay, wonderful. First and foremost, thank you very much for facilitating these calls. I realize how much of a burden they are. People present week on and weekend, and it continues to be an invaluable vehicle for us to ask questions as manufacturers of this year. My company had submitted an EUA earlier this year, which was granted authorization for a product on the condition that we perform a prospective clinical study post-authorization. In submitting our EUA, we've introduced a dual target design that ultimately we plan to submit for authorization as an improved assay design. Understanding Dr. Senzel's comment from earlier in today's call, clarifying that modifications can be implemented prior to authorization for laboratories performing high complexity tests and to utilize a least burdensome approach, will the FDA consider allowing us to conduct the studies for the improved dual target design rather than for the currently authorized single target design to support the change to the original authorization and avoid conducting tests as required?
Yeah, that's a useful question that the reviewer is a little bit too detailed for me to get into what test, what market study, and the timeline for that. Obviously, if our reviewers had asked for a post-market study, it was to confirm something so that the test that's currently on the market is known to be functional. So, and getting that result. So, but I think that's a the lead reviewer for your first authorization, and to if you submitted a PUA or you're preparing an EUA for the second test, that you include the reviewer that might be assigned for that. But I think we're going to want to know a lot more details before we give you specific advice. I don't want to say no without knowing those details. We obviously want to be as flexible as possible.
Thank you, Autumn, for your question. Myself, Michael, please ask your question.
Callers should unmute. We are not able to unmute callers. So if you can unmute, and star 6 may work. It usually works on Zoom if you're just on the phone or your local.
Just that, we'll move on. Can you hear me? Yeah. Okay, good. I may be in the wrong spot, but I'm not a developer. Semi-retired customer service consultant. I have 2 grandchildren that live at home. I have a wife that works as a medical assistant. I work with Scouts, 60 youth in my group, and also my church has only 20 members. We are having problems. It seems like there are only 3 over-the-counter home COVID tests available to have emergency use authorization. And they're not available. You can't find them anywhere. My question is, I'm concerned about availability. Are there other tests coming soon that
Hey, what's up?
get approved? Because this is, right now, big times are like at the end of October. And I've got 2 kids in school, and I'm an elderly person with pre-existing conditions. I really need to know their status.
So, thank you for calling. We, in fact, have authorized 13 home tests. I believe at least 7 varieties of them are authorized for over-the-counter. We're very well aware of the Delta curve that the manufacturers are struggling to keep up with the demand. And the U.S. government is funding an increase in the production of those in order to try to alleviate this issue. On the other hand, we have authorized over 60, I might be over 70 home collection devices. So there's a variety of sources for those collection through various entities as well as online sellers that are authorized to do that. So always double check that they're authorized. And many are home collection devices, so no prescription is needed. The turnaround time is obviously a little bit longer than a test that you can perform at home, and that should be taken into account. But I certainly know of individuals who have pre-purchased those home collection devices that are reasonably affordable and have stash at home. So the other option that you have is to look at any of the national providers of testing services or local providers of testing services. And they may have some options for you as well. So the national reference labs, the biggest among them are Laboratory and Quest. There are other reference labs out there, and they each have home testing available as well. So you may want to check them out with the home collection device. Amazon has a home collection device, and I apologize to the manufacturers I don't have a complete list, but the complete list of home collection devices and home tests is on the FDA website. And we continue to highly prioritize tests that come in for submission for the home environment, whether it's a home test for the diagnosis of SARS or home collection for the diagnosis of SARS, so either in the home, antigen and molecular tests. For collection, it's usually molecular tests. And those remain our highest priority, and when you receive a submission that has is complete and provides the evidence that we know the test would be accurate, we have unfortunately had to deny authorizations of tests that are clearly not accurate, or in cases where we had evidence that we cannot created that they submitted. So and as soon as these home submissions come in and they look good, they are the very highest priority for us to review. And overall, 410 test authorizations on the FDA. So there are very specific, but I certainly understand the current situation, and it's unfortunate. And at least from the FDA perspective, by authorizing accurate tests as fast as we can, we are going to continue to do that.
Thank you. The next question is from Tian Yang. So please unmute your phone and ask your question.
Okay, thank you. Can you hear me?
Yes.
My question is when FDA authorizing the new OTC test kits, will the manufacturing volume and supply capacity be considered as an important part? Because we noticed that the ED and assess that the newest approved two test kits are not in the market after this approval. So, just wonder if volume and supply capacity will be considered.
Yeah, I can't speak to any specific tests, but yes, I mean we're not going to spend a lot of time on a very low manufacturing test. And having been on the other end on the test developer side, and the ability to ramp up production is sometimes a challenge. It does take some time. You're not going to necessarily want to ramp up production until you're getting EUA authorization. And also, the distributors that you might use, including online and retail stores that might have other tests, that EUA authorized before making room on the shelf. So there are, unfortunately, real world challenges still present to me in the authorization that the FDA has.
Our next question will be from Sahil. Sahil, if you could unmute your mic and ask your question.
Can you hear me?
Yes.
Okay, so hi, my name is Sahil. I was just wondering if you had any updates on therapeutics, because a lot of hospitals are running out of therapeutics, and so that's, I just wondered if there were any ways or any more therapeutics approved using emergency use authorization.
So, first part of the device center, and this call is largely focused on COVID tests and authorization of COVID tests. The centers that would authorize therapeutics and/or vaccines would either be vaccines are authorized by the Center for Biologics, and many of the therapeutics are authorized by the Center for Drugs. So, you may be able to reach out to them to inquire as to their that you ask your question.
Okay, thank you.
In fact, you can reach out to the Division of Drug Information, DEI. They answer any of those types of questions, including the COVID questions. With that, our next question is from Arizona. So I'm going to unmute you, so please unmute your microphone and ask your question. Move on to our next person, Greg X. So Greg, please unmute your microphone and go from there. Move on to Jack. Jack, thank you. Jack.
Hi, do you get me?
Yes, we can hear you.
All right. So, as FDA claimed for many times that the antigen home test, antigen, sorry, can you hear me?
Yes.
Yes. Just from calling to interrupt. So, antigen home test is the priority. So, may I ask how big is the team working on the EUA of antigen home test?
It's a sizable team, and they are currently prioritizing home tests and point of care tests, and I meet with them weekly, if not more. Well, I meet with that specific team weekly to go over the entire solution and prioritize the review. And then we also meet daily as a full team and management team and home team and asking the questions of the public so that we can move quickly. As I mentioned earlier, in sort of the reverse of what I said, so if a submission comes in and it's complete and review is any questions, it gets high priority. But the reverse is also true, that if the submission is not complete and we have questions, it is going to not be as high a priority as complete and all the information is there in a manner in which we can understand how the validation is done, and we can move forward on that. It's incredibly rare for any of the submissions to be fully complete. So even on the highest priority test submission, there is a fair bit of back and forth. And we're entirely dependent, once we've asked the question, on how fast that gets back and forth. So the recommendation that we would have is that make sure that the submissions are complete, they're well done, that 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 they generally follow our recommendations. That if for some reason our recommendations are not followed, that will entertain questions on our part. Some of those potentially could be mitigated with written justification for their changes. But the workload is still high. We're still getting more than 100 EUA a month. This far into the pandemic. So, and as I mentioned at the top of the call, for those for a large number of clinicians now, they are roughly at the same level of priority, and we are moving to direct at least for the and move to the first in, first out. So those that have been waiting longest for a decision.
Right, thank you.
So with that, we have one more hand raised. Remember, if you want to ask a question, please raise your hand. We'll go to Greg S. and try again. We'll try again.
Please unmute your line if you wish to speak. We will designate you as a caller and call your name. If you haven't tried star 6 on your phone, calling in by phone or that, you're connecting to the internet and mute button. You are shown as muted, and you cannot give you the permission to. So there you go. Oh, children, you were unmuted for a second, and then you're muted again.
So please unmute yourself if you want to ask a question. Also, Greg asked, and I do unmute you, is telling me that you're using an older version of Zoom. So it's not allowing you, that may not be allowing you to unmute.
But that may be the situation. That's a good reminder of call. Maybe make sure you have the current version of Zoom. And if you're not able to get through the technical issues, you always have the email address for the upcoming town hall to ask the question ahead of time. But we also have the EUA template email address, and you can send us an email question there, and we'll endeavor to get back to you.
With that, it doesn't look like we have any other additional questions, so we'll move to that and try and I'll give you everyone one last chance. Please raise your hand if you have an additional question and you're available to unmute and ask that question. Sounds like we have no additional takers because, as was previously mentioned, if you do have a question, feel free to email us that question ahead of time, and we'll meet again in another 2 weeks. So thank you, everyone. We greatly appreciate your participation. We greatly appreciate the expertise of both Tim and Christian. Today's presentation transcript will be available at CDRH Learn. Please visit CDRH Learn at www.fda.gov. Train CDRH Learn. Note that we've updated the title of this section to make it easier to navigate. You'll now find the recordings in the subsection titled Coronavirus COVID-19 Test Development and Validation Virtual Town Hall Series. For additional questions about today's presentation topics, please send an email to CRH EUA templates at fda.hhs.gov. As we continue to hold these virtual town halls, we appreciate your feedback about the program series. So please complete a brief survey which you can find at www.fda.gov/CRH11. Finally, as a reminder, we are switching to a bi-weekly format, so please join us for the next webinar on September 22nd. This concludes today's town hall. Thank you.
The recording has stopped.